Hello everyone, this video is about cell-mediated immune response, NK and NKT cell function and mucosal immunity part 1, and this particular study note is developed by EPG Pathshala team, Government of India, MHRD. So I would like to express my sincere gratitude to the development team of this particular study note. Thank you so much. Host defense system protects from deadly diseases either through first line of defense, non-specific innate system, or via specific adaptive immune response. Various control mechanisms are employed for the tight regulation of immune system. These mechanisms maintain the homeostatic state of the immune system after the antigen has been eradicated. Several aspects of host defense system are synchronized by enormous ways, counting antibody, pathogen, and immune cells. Effector cells or mediator cells play critical role in modulating immune response. Progenitor cells are derivatives of stem cells. They can be lymphoid or myeloid derivatives committed to a particular cell lineage without self-renewal ability, unlike stem cells. T cells and B cells are natural killer cells are generated from lymphoid progenitor cells. Many different tissues and organs of the body constitute the immune system, which are classified into two major groups based on functionality of these organs, namely primary lymphoid organs and secondary lymphoid organs. Lymphatic system and blood vessels play a vital role in connecting these organs and thus forming a composed functional system as a whole. The body gets protection from the tumor growth and other infections via various independent functional cells which comprise the immune system. A non-specific immune response has been elicited by the white blood cells, which play imperative role in cytokines production and antigen presentation, while specific immune recognition and response are attributed to lymphocytes. History. Antibody-dependent cell-mediated cytotoxicity was discovered accidentally by Erna Moller in 1965. So ADCC is an accidental discovery. The natural killer cells were discovered as lymphocytes in mice by Ronald Herbermann and group in 1973, and the term is coined by Rolf Kissing and his colleagues in 1975. Hans Wigzell 1980 specified the role of natural killer cells in tumor cell killing. Ralph Bed and group and R.J. Folks group independently in 1987 revealed CD4 plus and CD8 plus lymphocyte population expressing NK and T lymphocytes features. NKT lymphocytes. Olivia and Albert 1994 revealed that NKT cells express invariant TCR in mouse and humans. Albert 1995 evidenced that NKT cells recognizes antigen presenting cells APCs via CD1 molecule. Quano et al. 1997 identified glycosylceramide glacer is a ligand to NKT TCR lymphocytes. Expressed in Latin words as lympha as water and cyte as container, lymphocytes are only cells in the immune system with specific immune response and developing memory. Three major lymphocytes subpopulation which constitute 20% of the white blood cell population are B cells, T cells, and natural killer cells. These subpopulation cells are morphologically similar but play different functions. B lymphocytes and T lymphocytes. The maturation place for B lymphocytes is bone marrow. The only cell in the immune system which is capable of producing antibodies are B cells. These cells express membrane-bound antigen binding receptors or antibody molecules which recognize antigen molecules and thereby leading to rapid division of cells to progeny cells which later on differentiate into plasma cells and memory cells. The site of maturation for T lymphocytes is thymus, thymic education maturation process. T lymphocytes also have antigen binding receptors on its membrane known as T cell receptors, but these are not antibody molecules. Antibody molecules on B cells can bind to the antigen alone, but receptors on T cell recognizes antigen only via major histocompatibility complex MHC molecules mediated antigen binding and even known as antigen presentation. A glycoprotein of cell membrane is recognized by the T cell known as MHC molecule, categorized as class 1 and class 2 MHC molecule. So, The site of maturation of T lymphocytes is thymus via thymic education maturation process. T lymphocytes also have antigen binding receptors on its membrane known as T cell receptors, but these are not antibody molecules. Antibody molecules on B cells can bind to the antigen anole, but receptors on T cells recognizes antigen only by a major histocompatibility complex molecules mediated antigen binding and even known as antigen presentation. A glycoprotein of cell membrane is recognized by the T cells known as MHC molecule, categorized as class 1 and class 2 MHC molecule. The two major populations of T cells are TH, T helper cells, and TC, cytotoxic cells, which can be differentiated on the basis of presence of CD4 and CD8 glycoprotein. CD4 and CD8 is glycoprotein on cell surface. Another subpopulation of lymphocytes is called natural killer cells or NK cells. These cells are born killers, as these cells without any prior antigen stimulation have the potential to kill via lysis different tumor cells as well as virus infected cells. These lymphocytes, unlike T cells and B cells, does not express any receptors, either membrane bound or for antigen, and that's why also known as null cells. Antigen, B cell receptor, B cell. Activated B cell, MHC2, cytokine, CD4, helper T cell receptor. Natural killer cells or null cells. Null cells were accidentally discovered in 1976 by immunologists while working on tumor-specific cells in mice model. Investigators have revealed a population of large granular lymphocytes which are non-specifically competent to homicide tumor cells. These lymphocytes are described as natural killer cells or null cells. Natural killer cells. A large granular and non-specific tumor killing cells are called natural killer or null cells. These cells are known as flight or fight cells. They does not produce memory cells and are non-phagocytic in nature. Unlike T cells or B cell lineage, these cells don't express surface receptors such as T receptors for antigen presentation and membrane-bound molecules like antibody molecules. NK cells share a common progenitor with the T cells and also express some common markers on membranes as by T cells. Despite the similarities with the T cells, NK cells does not mature in thymus. NK cells do not go through receptor genes rearrangement like B cells and T cells and do not express antigen specific receptors. NK cells mediated target cell recognition is not MHC class 1 and MHC class 2 restricted and neither has it generated immunologic memory. This cell constitutes 10% of recruiting lymphocytes in the peripheral blood. NK cells play a critical role in immune defense of host against virus infected cells and tumor cells as they are capable of producing important cytokines for immune regulation, for example. So, NK cells act earliest on infected cells with virus by inhibiting their activation, replication, and differentiation into functional cells, and thus act as a first line of innate immune response when life-threatening virus infection initiates. So, NK cells play critical role in immune defense of host against virus-infected cells and tumor cells as they are capable of producing important cytokines for immune regulation, for example, Th1 cytokines, specifically IFL. Although NK cells do not express membrane-bound antibody molecules or T cell specific TCR, but do express different surface molecules in order to interact with other immune cells. Some of these surface markers include CD56, which is an adhesion molecule, KIR, killing inhibitory receptors, and KAR, killing activatory receptor. CD16, FC gamma R3, also expressed on other effector cells such as monocytes, macrophages, etc. Other markers are there. Some of these surface markers are there. So, NK cells do not express membrane-bound antibody molecules or T cell specific TCRs, but do express different surface molecules in order to interact with other immune cells. Some of these surface markers include CD56, which is an adhesion molecule, KIR or killing inhibitory receptors, and CAR, killing activatory receptor, and CD16, also expressed on other effector cells such as monocytes, macrophages, etc. Other markers are shown. Killing activatory receptor, CD38, CD16, CD56, KIR, killing inhibitory receptor. killing activator receptor and killing inhibitory receptor. Mechanism of cytotoxicity. Missing self hypothesis. Scientists in 1990 proposed missing self hypothesis. His colleagues hypothesized that NK cells detects missing self, a class 1 MHC molecule, and lyse the aberrant cells. This mechanism plays an important role in recognizing and differentiating between self and infected cells. Natural killer cells are capable of killing tumor cells as well as virus-infected cells prior to cytotoxic T cell appearance. These NK lymphocytes differentiate between healthy self-host cells and infected or aberrant cells by two major ways depending on recognition of these cells by their surface molecules. One way include killing inhibitory receptors and another way is through CD16 Fc gamma R3 molecules. On binding to NK cells to healthy cells of host, KR receptors of NK cells bind to MHC class 1 molecules conserved region exhibiting self-peptide of host, which give a negative feedback to NK cells, thereby preventing the activation of NK cells regulating cell lysis. However, on any kind of virus infected or tumor cells, the MHC class 1 molecule showed downstream expression on the cell surface, because of which the KAR receptors of NK cells cannot bind with the altered cells, thereby activating the downstream cascade of NK cell mediated cellular toxicity of non-self or aberrant cells. The mechanism of cytotoxicity is based on production of granules from these large granulocytes or NK cells. These secreted granules kill the target cell through apoptosis or perforin-mediated lysis of altered cells. Once again, any kind of virus infected or tumor cells, the MHC class 1 molecule showed downstream expression on the cell surface, because of which the KAR receptors of NK cells cannot bind with the altered cells, thereby activating the downstream cascade of NK cell mediated cellular toxicity of non-self or aberrant cells. Here I have got some doubts. So I'm going to read this. Important role in recognizing and differentiating between self and infected cells. Natural killer cells are capable of killing tumor cells as well as viruses infected cells prior to cytotoxic T cell appearance. These NK lymphocytes differentiate between healthy self-host cell and infected or aberrant cells by two major ways depending on recognition of these cells by their surface molecules. One way include killing inhibitory receptor and other way is through CD16 molecule. On binding of NK cells to healthy cells of host, KAR receptors of NK cells bind to class 1 MHC molecule conserved region exhibiting self-peptide of host, which give a negative feedback to NK cells, thereby preventing the activation of NK cells regulating cell lysis. However, on any kind of virus infected or tumor cells, the MHC class 1 molecule showed a downstream expression on the cell surface, because of which the KAR receptor of NK cells cannot bind the altered cell, thereby activating the downstream cascade of NK cell mediated cellular toxicity of non-self or aberrant cells. The mechanism of cytotoxicity is based on production of granules from these large granulocytes. These secreted granules kill the target cells through apoptosis or perforin-mediated lysis of altered cells. Diagrammatic illustration of missing self hypothesis. Class 1 MHC perforance. Normal self cell. Class 1 MHC. KIR. No activation. NK cells. KIR activated, but cell is not activated. Healthy target cell, but infected non-self cell. Class MHC. Empty KIR. KAR activated NK cells. Target cell apoptosis will be taking place. Antibody dependent cell mediated cytotoxicity or ADCC, which is an accidental discovery we mentioned earlier. So macrophages, natural killer cells and other lymphocytes, the BBCs, neutrophils, eosinophils and monocytes are the effector cells causing cell-mediated cytotoxicity. When antibody-coated target cells are killed by these effector cells by binding to the antibody's Fc region and lysing the target cell in a non-phagocytic manner, this phenomenon is known as antibody-dependent cell-mediated cytotoxicity. When antibody-coated target cells are killed by these effector cells by binding to the antibody's Fc region and lysing the target cell in a non-phagocytic manner, this phenomenon is known as antibody-dependent cell-mediated cytotoxicity. Cancer cell target cancer antigen, antibody, Fc receptor, NK cell, perforin and granzyme released. The number of cytotoxic cells binds with the antibody molecule as they possess membrane-bound receptors specific for the carboxyl terminal end of IgG molecule termed as Fc region of antibody and expressed as CD60. When targets are attached to antibodies, these receptors bearing cells binds with the antibody's Fc region and thus leads to lysis of the antibody-coated target cells. This indirect method of targeting and killing target cells where the cytotoxic cells are non-specific by themselves, but where a specific antibody attached to target cells gives a specific targeting response. This process is referred to as ADCC. ADCC-assisted killing of target cells involve different mechanisms of cytotoxicity, but does not involve complement-mediated lysis. When the cytotoxic cell binds to antibody-bound target cells, their metabolic activity goes more activated, leading to increase in the cytoplasmic lytic enzymes such as lysosomes and granzymes. Perforin is a 70 kilodalton protein secreted by T cytotoxic and NK cells, which forms pores in the target cell. Perforin also controls lymphocytes proliferation. Perforin definitely leads to lymphohistiocytosis in mice. NK cells' mechanism of toxicity is very much similar to T cell mediated apoptosis of target cells. NK cell cytoplasmic granules contain perforin, because of which the target cells undergoes perforin mediated cytotoxicity. The target cells are lysed by the action of these released lysin lytic enzymes at the site of Fc mediated contact along with the secretion of tumor necrosis factor TNF from the cytotoxic cells such as NK cells, macrophages, activated monocytes, etc. These cytotoxic cells release pore-forming lethal proteins perforin and enzyme granzyme B, which activate the caspase cascade in the target cell, resulting into destruction of cells via apoptosis. So mice that lack NK cells has an autosomal mutation known as PAGE, which makes this model more susceptible to tumor growth after live tumor injection. Shirk-Higashi syndrome in humans. It's a recessive and autosomal disorder, has impaired effector cells such as NK cells, macrophages, etc., and was susceptible to lymphomas. Thus, we can conclude that NK cells play a critical role in defense against tumor growth. This is a diagram illustrating the mechanism of NK cell cytotoxicity on tumor cells. Antibodies bind antigens on the surface of target cells. NK cell CD16 Fc receptors recognize cell-bound antibodies. Cross-linking of CD16 triggers degranulation into a lytic synapse. Tumor cells die by apoptosis. NKT cells. There is a growing recognition of a heterogeneous group and a distinct lineage of T cells known as NKT cells, sharing some common surface markers with NK cells or null cells, and like T cells possess invariant alpha beta T cells receptors. These cells constitute 0.1% of total recruiting T cells in the peripheral blood. Basically, they differentiate distinctly from T cell, therefore differ from conventional T cells CD4 plus and CD8 plus, which in presence of class 1 and class 2 peptide MHC molecule recognize antigens. NKT cells have T cell-like receptors which in presence of class 1 MHC like molecule CD1 binds with the self and foreign glycolipid antigens. Variable levels of receptors which are typical of NK cells, especially CD16 and CD56, are expressed on NKT cell surface. On antigenic stimulation, a population of NKT cells secretes cytokines in large quantity, which is required for the B cell-induced antibody production and inflammation, and also for cytotoxic T cell development and expansion. This response system is required for early health when the development of conventional Th cell is still under progress. NKT cells and CD1D restricted and act as an immune bridge between adaptive and immune response of the host defense system. NKT cells are critical mediators in tumor immune surveillance and host immune response. NKT cells are limited in diversity and their populations consist of morphologically similar subpopulations, but they are functionally different, characterized on the basis of their surface markers, tissue location, effector functions, and TCR usage. Two major subsets based on the difference of TCR rearrangements is classified as invariant type 1 NKT cells and type 2 NKT cells. Another minor subset of NKT population is NKT like NKT11 and NKT1 minus, which contrast functions with the contrast functions. The two essential subsets of NKT cells acting on tumor immunity and cross-regulating the innate and adaptive immunity are variant type 1 and type 2 NKT cells. It includes innate and adaptive immunity are variant type 1 and type 2 NKT cells. It includes interacting with CD1D positive antigen presenting cells of the innate immune population, such as activated monocytes, dendritic cells, macrophages, and natural killer cells, as well as adaptive immune cells populations along with myeloid lineage, such as B cells and CD4 and CD8 positive T cells. NKT cells in an innate fashion reacts quickly and antigen stimulation by secreting cytokines in large amount, while directly or indirectly but rapidly triggering the adaptive and innate immune system. Thus, NKT cells act as an imperative bridge that modulated the major immune defense system of the host. NKT cells share both characteristic features like immunosuppressive as well as enhancing the immune response. We will be discussing the immune enhancing role of NKT cells at first. Receptors of NKT cells. NKT cells possess receptors common to natural killer cells or null cells as well as T cells and also common to other immune cells in the host immune system. Some common receptors expressed on their surface are as follows: CD1 or ITACR, semi-invariant TCR, then CD27 and OXO40 to interact with dendritic cells, CD40 along with CD1 to interact with B lymphocytes, CD1 and others to interact with antigen presenting cells or APC, then CD16 and CD56 assemble to NUN cells or NK cells, then NK 1.1 CD161 etc. Semi-invariant TCR, CD40L, CD27, OX40, CD16, CD56, CD40, NK 1.1, CD16, CD70, CD1D, OX40L. Mechanism of NKT cell toxicity. NKT cells are important mediators in regulating the immune response, especially type 1 NKT cells, attributing to its enhanced tumor-specific immune responsive route. These cells, either through the activation of other immune cells, can indirectly kill the tumor cells or can directly lyse the tumor cells. The direct cytolytic activity of these cells employ perforin-mediated killing and granzyme B-dependent mechanism for potential tumor cell death. These tumor cells, which express CD1D markers on their surface, are more susceptible for direct cell killing by NKT cells. Although NKT cells are capable of direct lysis of tumor cells, various studies revealed that anti-tumor activity of NKT cells is mediated largely through the activation of adaptive and innate immune system, primarily initiated by the Th1 cytokine cascade. An unusual glycolipid recognized by NKT cells are beta-galactosylceramide and beta-glucosylceramide, glucocerebroside, and respond strongly to alpha glacer, which leads to potent tumor rejections. Alpha glacer potentially activates type 1 NKT cells and leads to plentiful amount of IFN gamma production required for protection against tumor growth and thus revealed an anti-metastatic property. NKT cells contribute to tumor immune enhancing potential of host defense system by activating endogenous IL-12 pathway and early IFN gamma pathway. Even in the absence of external antigenic stimulus, NKT cells can potentially act against tumor growth through different subsets of its cell population. NKT cells on antigenic stimulation secrete IFN gamma in abundance amount, which further activate antigen presenting cells such as DCs, dendritic cells, and CD8 plus T-man cells. The interaction is assisted by the complex formation between TCR and CD1D, and also through CD40L and CD40 between NKT cells and DC. The activated DC promote adaptive immunity as they cross prime with CD8 plus T cells by the co-stimulatory receptor upregulation. This disease further stimulates NKT cells, other T cells and NK cells by the production of IL-12 and leads to the production of more IFN gamma. IL-12 and IFN gamma production drastically activates the effector cells population downstream. Both type 2 majorly and type 1 NKT cells directly lyse the tumor cells through perforin and granzymes B dependent cascade and extensively impact the production of CD8 plus T cells through the activation of memory CD4 plus T cells. NKT type 1 cells, NK and CD8 plus T cells directly lyse the tumor cells. So type 1 NKT cells also regulate the function of type 2 NKT cells. An NKT cell recognizes Mycobacterium glycolipids, which causes tuberculosis. And NKT cells has been employed for cancer treatment and vaccine development. NKT agonists have been used as adjuvant in vaccines. A nanovector containing CD1D stimulating NKT cells is a very recent development for effective vaccine production. So illustration of mechanism of apoptosis in tumor cells mediated through NKT cells. IFN gamma, type 1 NKT, CD4 plus T cells, type 2 NKT, CD8 plus T cells, tumor lysis, IFN gamma, tumor lysis, tumor cells, tumor lysis, NK cells. Now let's have a summary. The lymphocytes are truly crucial for the regulation and modulation of immune response in host defense system. Cells of the immune system are functionally interdependent by the production of broad range of cytokines. These cells negatively or positively through feedback mechanisms can regulate immunological functions. This can either boost or suppress the host immune system, broadly dividing the interacting immune cells of the defense mechanism. We consider lymphocytes and antigen presenting cells as important mediators. Lymphocytes can be B cells, T cells, and NK cells of lymphoid lineage and WBCs, neutrophils, eosinophils of myeloid lineage. So NK cells are bone killer cells, also known as fight or flight cells. These effector kill cells express surface molecules like CD16, which binds to the Fc region of the antibody molecule CD56. These cells kill the tumor cells through antibody-dependent cell-mediated cytotoxicity or ADCC. NK cells bind with the antibody-coated target cells and through perforin and granzyme-dependent killing of target cells, also known as ADCC. NK cells differentiate between self and altered cells via missing self hypothesis, where KIR receptors present on NK cell surface binds with the expressed self-peptide MHC class 1 molecules and self-healthy cells. NK cells does not get activated, leaving behind the self cells. Infected or apparent cells do not express MHC class 1 molecule on surface, because of which KIR does not bind with the altered self, leading to activation of NK cells, ultimately killing the target cells through perforin-mediated lysis or apoptosis. Other lymphocytes, NKT cells development site is thymus, where they randomly generate invariant T cell receptors and CD1 receptors after deviating from the T cell lineage. These NK T cells share some common receptors to natural killer cells such as CD16 and CD56, and some with T lymphocytes such as semi-invariant alpha beta TCR. Like lymphocytes expressing class 1 and class 2 MHC molecules, NKT cells express MHC class 1 like molecule CD1. So NKT cells are early responding cells when the Th1 T helper cells are still undergoing development. NK cells binds with the glycoproteins, especially alpha glacia presented by the MHC class 1 like molecules, and like peptide presenting MHC molecules of T lymphocytes. A broad range of cytokines, especially IL-12 and IFN gamma, are produced by these NKT cells, thereby leading to stimulation of innate and adaptive host immune system. Directly through perforin-mediated and granzyme-dependent cytotoxicity or directly by triggering CD8 plus T cells and CD4 plus T cells and antigen-presenting cells, APCs, NKT cells leads to tumor cell apoptosis. An NKT cell is critical for autoimmunity suppression and rejection of grafts and also resists infection along with promoting immunity against tumor growth.